Okay, so welcome back to this next video in which we are discussing multi-drug transporters. Okay, so we've now looked at ABC multi-drug transporters now. We've seen many different examples of ABC multi-drug transporters. We're now going to go through the other four families of multi-drug transporters, and don't worry, it won't take us as long this time because each one of them, I've only got one example that we're going to discuss of each of them, and we'll have a look at the mechanism by which they move these substrate molecules out of the cell. Okay, right. So the next family then that we are going to move on to is the RND family. Okay, which remember stood for the resistance nodulation division family of transporters. So the RND transporters is up next. Okay, now before we actually start, let me just say that we have now completely exhausted the supply of primary active transporters that are multi-drug transporters. The ABC transporters is the only family of multi-drug transporters that contains transporters that are actually active transporters that are primary. Okay, all the other multi-drug transporters that are not in the ABC transporter family are going to be secondary active transporters, so their movement of these substrate molecules out of the cell is going to be coupled to the movement of something else down its electrochemical gradient. Okay, right, and this is going to be true of the RND transporters. Okay, so let me give you the example, the key example. There is a very, very famous example of an RND transporter, okay, and this is what's known as ACRA, ACRB in E. coli. Okay, so this is my main example of an RND transporter. Okay, so this is going to be a secondary active transporter that is actually going to use the proton gradient across the inner membrane of E. coli. Okay. In addition, it is going to actually be part of a much bigger complex. Okay, and this much bigger complex is something known as ACRAB Tol-C. And we will come on to the structure of this complex in just a moment. Okay, before we go over what this complex is, I firstly want to tell you a little bit more about the structure of E. coli, okay, because it's going to be necessary to understand this transporter. Okay, so let's study E. coli in a little bit more detail. So E. coli, Escherichia coli in full, might as well write that out now I've said it. Escherichia coli is its full name. Okay, it's a gram-negative bacillus. Okay, so it's a rod-shaped or bacillus bacteria. Okay, and it's gram-negative. So if you stain it with the gram stain, it stains red. Okay, now all gram-negative bacteria generally have two cell membranes. Okay, an inner membrane and an outer membrane, and then in between these two membranes, they then have a very small portion of cell wall. Okay, and this is the portion that will then stain red in the gram staining process. Okay, right, so let's put some labels on this picture. So the membrane that's the inside membrane, this is what you would call the cell membrane. Okay, it's also called the inner membrane. Okay, so this is the one that actually surrounds the cytoplasm of the cell. Okay, so this is the inner membrane. Okay, then this outer membrane, this will be called the outer membrane. Okay, and then there's a space in between the inner and outer membrane, and that has a special name. The space in which the cell wall is located is what's known as the periplasm. Okay, similarly to cytoplasm, which is inside the cell, or it's also called the periplasmic space to be fancy. Okay, right. Now, bacteria do not have mitochondria. Okay, mitochondria are believed to have arisen from a bacteria cell being engulfed by a precursor to a eukaryotic cell. Instead, the electron transport chain that is operating within bacterial cells is on the inner membrane. Okay, so their respiratory chain process is on the inner membrane, and what happens is they pump protons through the electron transport chain proteins from the cytoplasm of the cell into that periplasmic space. So if I draw a bigger picture here, Okay, let's say this is the inner membrane here, which I'll abbreviate to IN, and let's say this is the outer membrane, which I'll abbreviate to ON. Okay, and then this is the cytoplasm in here. The respiratory chain, the electron transport chain, will move protons from the cytoplasm into the periplasmic space here. So remember, in the electron transport chain, those electrons are gradually moved along those complexes, and they'll gradually give up energy, and the energy is used by the complexes to pump protons from one side of the membrane onto another. Okay, that's the same as what happens in the inner mitochondrial membrane, only here it's happening in the inner membrane of E. coli. Okay, and what this results in is a very powerful electrochemical gradient favouring the movement of protons from the periplasmic space to the cytoplasm. So basically you end up with absolutely loads of protons in the periplasmic space and very few in the cytoplasm. Okay, so there's a big concentration gradient across the cell membrane. Okay, in addition, you end up with an electrical gradient across the cell membrane because these protons that you are moving, they have positive charge. So when you're moving the protons into the periplasmic space, you're increasing the electrical potential here. Okay, so the electrical potential of this periplasmic space, which I'll abbreviate to capital E for electrical potential and then P for periplasmic space, this is going to be going up when you move those positively charged protons there. Okay. Meanwhile, the electrical potential in the cytoplasm, which I abbreviate as EC, when you're moving protons out of the cytoplasm, you're moving positive charge out of the cytoplasm, the electrical potential in the cytoplasm is going to go down. That's going to create a very powerful electrical potential difference across the cell membrane. Okay, so now you're going to have a voltage, basically, or an electrical potential difference across the cell membrane. So basically, if we look at the voltage or electrical potential difference from the periplasmic space to the cytoplasm, okay, so the electrical potential difference that you undergo if you move from the periplasmic space to the cytoplasm, that's going to be negative, basically, because now the periplasmic space's electrical potential is high. Okay, so we'll use a little diagram like this to show this. Here's the electrical potential of the periplasmic space. Here now is the electrical potential of the cytoplasm. So when you move from one to the other, you go down in electrical potential, and that means that there is an electrical potential difference across that inner membrane. Okay, and that's a voltage, so it will be negative, basically. If you move from here to here, you go down in electrical potential. Okay, now, protons want to be where the electrical potential is lower. They're positively charged ions, okay? They want to go where the electrical potential is lower. So that's currently the cytoplasm, so they're going to want to go to the cytoplasm, basically, okay? So not only is there this concentration gradient, but there's also this electrical gradient, and therefore there is a strong electrochemical gradient, which is the combination of the electrical and the concentration gradient, favouring the movement of protons back into the cytoplasm across the inner membrane. Okay, and this is going to be what this ACRB multi-drug transporter is going to use to acquire energy to move substrate molecules out of the cell. Okay, so without further ado, let me show you the ACR-AB Tol-C full assembly. Okay, so it is sitting in the periplasmic space. So let me redraw this picture out again. So here is the outer membrane, here is the inner membrane. Okay, so in fact I'm going to go over to drawing the membranes as phospholipid bilayers. Okay, so I'm promoting the membranes up to being shown, instead of just as one line, to being shown as two lines. Okay, representing the inner and outer leaflets of those membranes. Okay, now, basically, ACRB, the actual multi-drug transporter here, is present in the inner membrane, and it's present as a trimer. Okay, so I'll put it here. So here it is, the massive great thing, made up of 3 separate proteins, all trimerized together. So all of these are ACRB proteins. So these are all ACRB proteins. Now let me just show you the membrane spanning topology of ACRB proteins then. So if we draw this inner membrane again, okay, so I'm just taking one of these individual subunits of the ACRB trimer out here, and we're going to have a look at its structure. Okay, its structure looks like so. Okay, it's got, again, 12 membrane-spanning alpha helices. Now, its amino terminus will start in the cytoplasm, and then it will have, keep it off, a very large domain that's in the periplasm, in between the first membrane-spanning alpha helix and the second, and then the other loops will be much smaller. So this loop between the first and the second membrane-spanning alpha helices, that's going to be huge, and that's going to make a huge periplasmic domain, basically, which is going to be important in the function of the transporter. Okay, so here is our first periplasmic domain here. Okay, then that's the first cluster of 6 membrane-spanning alpha helices. Now we've got the second one. Now again, between the 7th and the 8th membrane-spanning alpha helix, you have a large periplasmic domain there. Okay, so this is another large periplasmic domain, which will be making this portion that is within the periplasmic space here. Okay, right, so this is the structure then of a single ACRB monomer. Okay, so you take 3 of these things, trimerize it together to make an ACRB trimer, as is shown here. Okay, right, now, to make the full ACR-AB Tol-C thing, what you need to do is you need to join this to an ACR-A protein and a Tol-C protein. So let me first explain what these two things are. So I'll start with Tol-C. Tol-C is effectively a tube that crosses the outer membrane. So it's a tube through the outer membrane. So this here, this is the Tol-C. Okay, so I'll colour this in blue here. So this is Tol-C protein here. Okay, a tube through the outer membrane. Now, finally, ACR-A is a ring that is believed to connect Tol-C and the ACR-B trimer down here. Okay, so it's supposed to go around the whole Tol-C up here and the whole of the ACR-B trimer down here. Okay, so let me colour this in. So this is the ACR-A protein, which forms this ring connecting the Tol-C tube to the ACR-B trimer below. So this is the ACR-A protein. So basically, this is the idea, okay? ACR-B, the trimer of ACR-B here, this is the active transporter, this is the multi-drug transporter that is going to be grabbing drug molecules that are in the periplasmic space and also the inner membrane here. And it's also potentially believed that it grabs them from the cytoplasm as well, okay? It's going to be grabbing drug molecules, it's going to be pumping them into this Tol-C tube here, okay, and then they've got no choice but to move out of the Tol-C tube out into the extracellular fluid over here. And that's how this entire configuration here operates to remove drug molecules. And this full thing is then called the ACR-AB Tol-C complex. Okay, right. So let's now talk then about how ACR-B actually functions then. Okay, so this is quite complicated. Firstly, we need to remember that it is a secondary active transporter, so it's going to be coupling the movement of drug molecules to the movement of protons back across the inner membrane. Okay, now let's begin this process then, and we're going to begin it by looking at a single ACRB monomer. Okay, so not at the moment the entire trimer, just a single ACRB monomer. Okay, so what I'm going to do is effectively take a little cross-section here of my ACRB monomer here. Okay, so here it is sitting in the inner membrane like so. Okay, and it would be involved in this trimer in reality, so there'd be 2 more sitting around it. But we're just now looking at a single one of these. Okay, basically what happens is it can be in 3 states. It can change conformation and be in 3 different states. Now in the first state, which I'll call state 1 here, it has a little binding site available at this sort of position that can accept drug molecules either from the periplasmic space or the inner membrane here. So remember, this is the inner membrane. Okay, so our hydrophobic drug molecules could be in the periplasmic space here, okay, or they could be in the inner membrane. Okay, so remember this is an extrusion mechanism for hydrophobic molecules, and we're specifically interested in drug molecules. Okay, so this then is in the access state at the moment, where we have this site that is available, access state. We have this site that is available for drug molecules to come and bind in from the periplasmic space or from the inner membrane. Okay, right. Then what can happen is it can change conformation, and I would like to get this all on one page. So I might have to draw it on this picture here, so I might have to do it all on one protein, all on one picture rather. Okay, so that's state 1, where you have this site open and available to the periplasmic space and the inner membrane. Okay, state 2 then, it changes conformation and it takes the molecule into the middle of the protein. So whatever is bound to this access site is now, you change conformation and it's now in the middle of the protein, in this binding site here. So this is state 2. And then finally it will change conformation a third time, and then it will move to an extrusion site here. Okay, so it will now be in an extrusion state in state 3. Okay, and in this state it will release the drug molecule into this pore that's in the middle of the ACR-B trimer here, and then it will have no choice but to go into the Tolsi tube and out of the cell. Okay, so across the outer membrane into the extracellular fluid. Okay, now it's more complicated than that, of course, because you have all 3 of these separate ACR-B proteins, and the idea is that all 3 of them are cycling in between being in these 3 states. Okay, and they're all at a different level of the cycle. Okay, so let me explain what I mean by that. So let's say this one closest to us, this is currently in state 1. Okay, so it's currently in the state where it has an open access site for a drug molecule to come in from the periplasmic space or the inner membrane and bind there. Okay, so what's going to happen is a drug molecule is going to come in and bind there whilst this one's in state 1. Meanwhile, this one here will currently be in state 2. Okay, so it will have a drug molecule already bound, but that will be in the middle of the protein at the moment. Okay, so it will be in that state 2. Then this one over here, right at the back, this will be in state 3 at the moment, and it will therefore have its drug molecule in this extrusion site now. Okay, so it will be in the extrusion state, and it will be letting go of its drug molecule and allowing it to go into the Tolsi tube. Okay, so all of these 3 ACR-B proteins in the ACR-B trimer are in different states of this cycle, basically. Okay, so what then is going to happen is each one of these is going to update now. Okay, so what's going to happen is a drug molecule will bind to number 1, the one back here, which is in number 3 state, that will let go of its drug molecule, and then what will happen is the whole thing will update, conformational changes all around. This one here will go into state 2, where the drug molecule that it had bound is now in the middle of the protein. This one will go into state 3, where it's extruding its drug molecule into the Tolsi tube. And this one that was in state 3 will now go back into state 1, where it can collect a new drug molecule from the periplasmic space or the inner membrane. Okay, right, now what drives that conformational change? Well, every time you undergo these 3 conformational changes in the 3 ACR-B proteins, you have to move a proton across the inner membrane. So a proton moving across the inner membrane seems to cause these 3 conformational changes in the 3 ACR-B proteins. And then it just goes through this cycle after cycle after cycle, and each one of these is then going to be moving drug molecules through. Okay, and you can see that the stoichiometry here is 1 proton per 1 drug molecule. Because remember, each proton is going to cause each one of these ACR-Bs to update by 1. Okay, but it's doing it for 3 of them. Okay, and there are 3 steps here. So each one of these has to go through 3 updates for a drug molecule to be extruded. So you might think, but hang on, it looks as though 1 drug requires 3 protons, but then you have to remember that the proton is updating all 3 of them, and therefore that cancels it out. So overall, the stoichiometry here is 1 proton for 1 drug molecule being extruded. Okay, so ACR-AB Tol-C then, it has this rather incredible mechanism of the ACRBs cycling between these three states and them all being at a different state of this cycle, and the updating of the state is driven by the movement of the proton across the inner membrane back into the cytoplasm. Okay, so as I say, at the present, what we know is that it can take drug molecules from the periplasmic space and also drug molecules that are dissolved in the inner membrane, because remember, these drug molecules are hydrophobic that are being extruded by this, so they could be in the inner membrane because that's a lipid bilayer after all. Okay, but we don't know at the present that it does take them from the cytoplasm. We think that it must do, but we don't yet know how its structure allows it to do that, okay? Because this access site doesn't seem to be anywhere near the cytoplasm. Okay, right. So, examples of drugs then that can be extruded by ACR-AB Tol-C. Well, two key examples are once again the drug doxorubicin. Okay, so doxorubicin, as well as being used as an anti-cancer drug, is actually used as an antibiotic. Now, it is not a first-line antibiotic, it is fair to say, okay, but it can be used if other antibiotics really are failing. Okay, so again, I'll remind you that it's a DNA intercalator. It sits in between DNA double base pairs and causes problems with DNA replication and transcription. And it does that just as well in bacteria as it does in cancer cells, and therefore stops the bacterial cells from dividing. Okay, another example of an antibiotic extruded by the ACR-AB complex is minocycline, okay, which is a tetracycline antibiotic. Now, all tetracycline antibiotics work by stopping protein synthesis, so they inhibit protein synthesis. So they stop the cells from being able to make any more proteins, and that also causes the cells to stop dividing. Okay, so both of these antibiotics are extruded by the ACR-AB Tol-C complex here. Okay, right. So let's now move away from the R&D transporters then, and let's look on to the MFS transporters, the major facilitator superfamily transporters. So MFS, our third category of multi-drug transporters. So we want to see an example from the MFS superfamily of a multi-drug transporter, and I should say that the major facilitator superfamily, its name is correct. This is a massive great family of proteins that are found in all different forms of life. Okay, so many of the transporters in this are not multi-drug transporters, they are just active transporters. Okay, so we're going to look at specific examples from this family which are multi-drug transporters. So major facilitator superfamily. Okay, so the example that we are going to look at is again from E. coli, okay, and it's the EMRD transporter. Okay, and later on we'll see another protein that's named similarly, EMRE, okay, but that's in a different family. Okay, now again, EMRD is an example of a secondary active transporter, and again, it's present in the inner membrane of E. coli, and it's going to use the proton gradient to extrude the drug molecules. Okay, so let me show this again. So here is the E. coli bacterium, that's the outer membrane. Here's the inner membrane. Okay, and EMRD is going to be present in the inner membrane like so, and it's going to use the proton gradient that is across the inner membrane in order to move drug molecules across the inner membrane into the periplasmic space. Okay, now it's not involved in these massive great complexes the way that ACRB was. Okay, it's just a single little transporter. Okay, right, so it's going to allow a proton to move back into the cytoplasm of the cell in exchange for a drug molecule moving out, and therefore it's a secondary active transporter. Specifically, it is a antiporter. Okay, right. Now I'm just going to show you the membrane spanning topology of EMRD, and then we'll call it there for this video, and in the next video we'll have a look at the actual mechanism by which EMRD works. Okay, so if I show you the inner membrane now as a phospholipid bilayer, so we've gone from drawing the inner membrane as a single line to now drawing it as two lines, so don't get confused, this is not the inner membrane and the outer membrane, this is the inner leaflet and outer leaflet of the inner membrane. Okay, and the membrane spanning topology of EMRD is 12 membrane spanning alpha helices, once again clustered into two bundles of 6 membrane spanning alpha helices. So here's the amino terminus and here's the C terminus. Okay, right, so we'll call it there for this video, and in the next video what we'll do is we'll have a look at the mechanism by which EMRD works, and then we'll move on to the two final families of multi-drug transporters, which are the SMR family and the MATE family.